Hi everyone, welcome back. We all know that human body consists of trillions of cells, and in order for them to function as a single unit, as a single organism, they need to coordinate among themselves. And how do we achieve that marvelous feat? Well, chemicals. It's chemicals. And we all know of chemical signaling methods, be it endocrine, as seen with most hormones, or it may be paracrine, as happens across synaptic clefts, or it may be autocrine as well. But today our topic is not about chemical signaling. Cells can and do interact without chemicals by physical means, by interacting with one another. So, without further ado, let's dive in. Sometimes it so happens that physical contact between one cell and another, or between a cell and extracellular matrix secreted by another cell, can transmit signal. And that's our topic for today, juxtacrine signaling. Let us take the example of adhering junctions serving the purpose of literally adhering one cell with another in a tissue. But do you know that they have signaling function as well? Here on the left side is one cell and on the right another. Before we can understand how adhering junction may participate in signal transmission, let us first understand what adhering junction is composed of. It is formed by interaction between integral membrane protein present in the opposing plasma membrane of two interacting cells. The name of this protein is cadherin, and their interaction is calcium dependent. Clustering of cadherin to form adhering junction causes a secondary clustering of a group of intracellular proteins called catenins, as shown here in this video. One such catenin is beta-catenin. The catenin in turn serves as attachment site for actin cytoskeleton. It is by such arrangement that adhering junction maintain normal cell architecture. Under normal circumstances, when adhering junction is intact, most of the beta-catenin is sequestered around the adhering junction, and number of free beta-catenin in the cytosol is very few. But what if the adhering junction is disrupted, say for example by stimulation of some growth factors? Well, once the interaction between cadherin disrupts, the secondary clustering of beta-catenin, as mentioned earlier, is also disrupted. These free beta-catenins now translocates to the nucleus, and there they act as transcription factors, which ultimately leads to cell migration and proliferation. And this is how adhering junction takes part in signal transduction by physical means, an example of juxtacrine signaling. There exists a protein by the name adenomatous polyposis coli that prevents rise of free beta-catenin in the cytosol. They phosphorylate free beta-catenin and targets them for destruction, as shown here in the diagram. There is a type of colon cancer that occurs due to mutation of APC, or adenomatous polyposis coli protein. This is because APC prevents abnormal rise of beta-catenin in the cytosol, and their mutation causes beta-catenin to rise in concentration in the cytosol, which ultimately translocates to the nucleus and causes cell proliferation and migration, giving rise to colon cancer. So far in this video, we have discussed that adhering junction can function as a means of juxtacrine signaling. But examples of juxtacrine signaling is not restricted to adhering junction alone. Let us take the example of gap junction, also known as connection. Incidentally, the name of my channel as well. And talking about my channel, if you like this video, don't forget to hit the like and subscribe button. And now let's get back to the topic. Imagine that these two circles here are two cells connected by gap junction. You know that during cell injury, plasma membrane loses its selective permeability and calcium enters the injured cell in huge numbers, further aggravating the situation. This calcium inside the injured cell may pass through the gap junction already present and injure a neighboring cell. But fortunately enough, cells have protective mechanisms in place. Rise in calcium concentration following injury closes off the gap junction, thereby preventing the adjacent cell from meeting the same fate. And that's another example of a physical connection taking part in juxtacrine signaling. Have you heard of such developmental events such as axonal guidance in nervous system and endothelial cell guidance in vasculature? Well, if you have not, don't worry. And I guess you have guessed it right already. It also involves juxtacrine signaling. So, let's learn about Ephrin and Ephrin ligand interaction. Let's suppose this is a cell membrane with a transmembrane protein by the name Ephrin ligand, and this is another cell membrane with another transmembrane protein called Ephrin receptor. And such juxtacrine interaction between Ephrin and Ephrin receptor is crucial for axonal guidance in nervous system and also in the guidance of vascular endothelial cell growth. So far we have discussed about involvement of adhering junction in cell signaling. We have also discussed gap junction taking part in juxtacrine signaling and also about role of Ephrin and Ephrin receptor as part of juxtacrine signaling. Apart from these, there are a few other more interesting examples coming up soon in my next video, and see you then. Bye.